up guys? We've completed the first two parts of the eyeball. Now let's sum up the topic of the eyeball by discussing few more important points left. Okay, the first point is about macula lutea. Yes, macula lutea is a yellow spot at the posterior part of the eye, just lateral to the optic disc. If we talk about exact position, inferior-lateral to the optic disc, macula lutea lies. So it means at the posterior part of the eye, there is a yellow spot inferiorly, slightly lateral to the optic disc, known as the macula lutea. Okay, there is a depression in macula lutea. The depressed part of macula lutea, actually, yes, the depressed part of macula lutea is known as fovea centralis. Yes, it is known as fovea centralis, and fovea centralis, it is an important feature in the eyeball. Fovea centralis, because it has the sharpest visual acuity. Yes, it has the highest and sharpest acuity of vision. That means that it can see objects very clearly, and even it can see the minute details of the object. Yes, very minute details of the object. And it is because fovea centralis, the depressed part of macula lutea, it is packed with lots of cones inside it. Rods and cones, I have told you, photosensitive cells of the eyeball. So fovea centralis contains only cones, and cones have the ability to see the details of the objects very clearly. So fovea centralis has the sharpest visual acuity. Now coming to the next point, that is limbus. This is actually the sclerocorneal junction. Yes, the sclera and cornea both are parts of the outermost coat of the eyeball. We have discussed already. So sclera and cornea, where they meet, that part is known as the limbus, the sclerocorneal junction. Commonly it is known as limbus, but we can also call it as the sclerocorneal junction, where sclera and cornea, both the outermost coat of the eyeball, meets. That is the junction of sclera and cornea. Also, okay, let me explain here. There is one more part in the limbus. Actually, the deep part of the limbus. In the limbus, there is also a canal, a circular canal, known as the canal of Schlemm. So, the deep part of the limbus, the sclerocorneal junction, there is a circular canal which is known as the canal of Schlemm. Yes, canal of Schlemm of the eyeball lies in the deepest part, or the deep part of the limbus, that is the sclerocorneal junction. So few more points which were left. We are done with the macula lutea. I hope आपको समझ आ गया होगा. फिर आपका sclerocorneal junction and canal of Schlemm. Moving on further, let me tell you the difference between the rods and cones. Photoreceptor cells, I have already told you that the neural layer of retina. Retina consists of the two layers, pigmented epithelium, neural layer. The neural layer consists of the photoreceptor cells. There is the rods and cones. So there are some differences between rods and cones. Let us study. First difference, there are over 100 million rods in the eyeball, and over 5 to 7 million cones in the eyeball. Especially these are in the neural layer of the retina. Next, rods are rod shaped in the eyeball, and cones are cone shaped in the eyeball, whereas few cones in the fovea centralis, in the macula lutea, which is packed with lots of cones, are rod shaped also. Then the most important point is rods have the photopigment, rhodopsin, and cones have the photopigment called iodopsin. Rhodopsin and iodopsin plays an important role in the mechanism of vision. Next point, it is that rods are more sensitive to light. Yes, it means that even in very small light or dim light, they can work and we can easily see through rods. Whereas on the other hand, cones are less sensitive to light. They need more and bright light for vision. Okay, more sensitive to light, these are rods, and cones are less sensitive to light. And on this basis only, rods are meant to function in the night time. Nighttime there is low light or dim light, so rods function in the night time, and this vision is called the scotopic vision. Yes, scotopic vision is a function of rods, whereas scotopic vision. On the other hand, if we see in the cones, as I have mentioned earlier, that these are less sensitive to light. So less sensitive to light, they are meant to function in the daylight, in the bright light, and that vision is called the photopic vision. Yes, so rods function for the scotopic vision and cones function for the photopic vision. Next point, rods are responsible for black and white vision. Through rods, we cannot see the colors or the colorful details of the object. And these are only responsible for the black and white vision. And whereas cones, these function for the colored vision. So yeah, cones are responsible. So we can see different colors. So these were a few differences according to the shape, according to the photopigments, rhodopsin. Also, rhodopsin contains a derivative of vitamin A. Yes, that is why it is said that we should take more vitamin A for our healthy eyes. Okay, coming to one more important point, if I tell you, before we get into the mechanism of vision, neural layer. So we have been talking about neural layer of retina before also. The outer coat of the eyeball, sclera, cornea, middle coat, and then the inner coat, retina. So retina contains the pigmented layer and neural layer. So there are cells in the neural layer. We'll discuss few things about them. Number one is the photoreceptor cells. Photoreceptor cells contains the rods and cones. I have told you the difference between rods and cones. Next neural layer contains the bipolar cells. Very important cells in conducting the impulses, bipolar cells. Next is the third type of cell in neural layer. It is ganglion cells. They also conduct the impulses and transfer them to the optic fibers or optic nerves. So neural layer consists of the cells photoreceptor cells, bipolar cells, and ganglion cells. Remember these names? So bipolar cells and ganglion cells, these both transmits the action potentials or impulses. Yes, they actually conducts the impulses, transmits the impulses to the bipolar cells, then to the ganglion cells, and further ganglion cells transmit these impulses to the optic nerves. And actually the bipolar cells and ganglion cells, they transmit the impulses or action potential through the optic nerves to the visual cortex. Yes, visual cortex is area of the brain to which the ganglion cells through optic nerves transfer the action potentials. So these were the cells in the neural layer: photoreceptor cells, bipolar cells, and the ganglion cells. Bipolar cells and ganglion cells, they transmits, that is transfer the action potentials to the visual cortex of the brain for vision, and photoreceptor cells, they generate the action potentials. Okay, now moving to mechanism of vision. It won't take much time. Let's start. Rhodopsin, its photopigment, it splits into two derivatives, its two components, that is opsin and retinal. So these two are the components of the rhodopsin, opsin and retinal. Opsin, it is actually a protein in the rhodopsin, and retinal, whereas retinal is an aldehyde of vitamin A. So through this dissociation, action potentials are generated in the photoreceptor cells, that is the rods and cones. Action potentials are then transmitted to the visual cortex area of the brain through bipolar cells and ganglion cells. Rhodopsin, actually it dissociates in opsin and retinal when light falls on the retinal layer of the eyeball. Yes, when lights fall on the retina, rhodopsin, it dissociates in the opsin and retinal. This dissociation causes the changes in the structure of opsin, and there in the neuronal layer the action potentials are generated in the photoreceptor cells, that is the rods and cones. Action potentials that is the same, we can also use a impulse for action potential. So एक बार फिर से रिवाइज करते हैं. Actually the mechanism of vision: light rays falls on the sclera, then the cornea, and then retina. So the neural layer has photoreceptor cells, rods and cones, and then the rods have photopigment called the rhodopsin. Rhodopsin, it splits into the opsin and retinal, and therefore produce action potentials, which are then transferred to the visual cortex area of the brain for vision. So these action potentials, which are generated in the rods and cones through rhodopsin, are then transferred to the bipolar cells, ganglion cells, and lastly through optic nerves at the blind spot to the visual cortex area of the brain, where neural impulses are analyzed and we can see the image which is actually formed on the retina through all this process. This was all about eyeball. Till then, keep learning, keep growing.